Hi everybody, my name is Esmond.
And I'm Sparshita.
We're here today at the RNT&E to conduct an interview as part of the UCL MSc blog. This blog was set up to curate interesting things in science and also to showcase the excellent work that UCL researchers are engaged in.
We will be interviewing Dr. Martin Birchall, who is a consultant ENT surgeon and also a clinician scientist. He was involved in the world's very first synthetic trachea transplant. So let's just go in and get to know what it's like to be him.
Cool.
Good morning.
Oh, good morning. Hi. Morning. Hi.
So good morning, Dr. Birchall.
Morning.
We would like to interview you for our MSc blog, and we would like to start by asking you a bit about your educational background and what was it that eventually made you get into clinical research?
So I went to school in Liverpool and I went to Cambridge University for medical school. And I guess when I came out of medical school, I found that there was so much we didn't know, particularly about my chosen specialty, which was ear, nose and throat, or otolaryngology. And the bits of my job that I was enjoying the most really were the ones where we were exploring new things and new ideas and trying to understand what the literature behind that was. I then progressed through training, and when I was in Australia finishing my fellowship there, I realised that I was mostly interested in the larynx and this wonderful enigmatic organ that allows us to talk and breathe and speak and swallow and all these human things, and yet how little we really understood about it and how to replace those functions if they go wrong. And so I decided to make that really the focus of the work I was going to do alongside my clinical work. So a combined research, clinical life, but focusing on the larynx. Really.
Martin has a whiteboard with all the projects he is involved in and the various stages they're in. Martin, how diverse are the people you work with, and what are some of the qualities that are important to leading a multidisciplinary team?
So in 2009 I moved to UCL because really this gave the opportunity to specialise in a very multidisciplinary environment. I always tell people that at UCL you can just walk down the road and put together a world-class team from virtually any discipline. And this is really important in regenerative medicine. It's really what's made it take off as a scientific and medical discipline right now, is the huge interdisciplinarity of it all. And so I work with consultants and with groups right across a whole range of fields. For translational science, you need to have some input from basic and discovery science, so-called blue sky science, at the beginning. But really we're working with ideas that have already been worked up in the laboratory to a large extent. Maybe we don't know everything about them, but we know enough about them to confirm their potential efficacy in what we call preclinical scenarios, which is either in small animals or larger animals, or in bioreactor models of biological systems. And we have all of these things in our group. Having worked those things through, you then have to develop the plans for clinical trials and perform early phase clinical trials. And to do that is very time consuming, very expensive, and you need an input from a whole range of other skills. So from the beginning we have some discovery science groups in epithelial biology and in tissue engineering science. Then we have groups working in the preclinical side who are made up of a combination of doctors who are training in science, postdoctoral scientists specialising in this field, but also engineers who have designed the bioreactors and designed the delivery devices that we use, material scientists, chemists, who produce new biomaterials which we can use as scaffolds. And then moving through, we then have scientists who are specialised in the development of early phase clinical trials, so statisticians, trialists. Really the science behind this is highly multidisciplinary and very important, but we mustn't lose sight of the fact that we're trying to develop treatments for patients, and so patients themselves form part of the team, and we have public and patient involvement groups who are involved in all aspects of our research to make sure that we do keep that focus and that the clinical trials address the questions that they really want. There are different styles of leadership. As you can tell from the description I've given already, this is a very disseminated group with lots of people doing lots of things in different places. And the best way to lead a group like that is facilitatory, especially in a place like UCL where you're working with very brightest people right the way up from MSc and PhD students through to the actual principal investigators themselves. These are all extremely intelligent, driven people. So long as they have common goals, you can blend teams and build teams within teams, but it has to be facilitatory, as well as aiming to our main goals, which are development of new treatments. Our secondary main goal is to develop the careers of these people so that they can actually be considerably better in the long run at delivering treatments than we are at the moment.
Dr. Birchall, I have always been curious about this. When you introduce a new therapy, say in case of clinical trials, there's always an uncertainty whether the therapy would work or not. So at what point in preclinical research do you actually decide that you have to go ahead with the clinical trial? And how do you deal with this whole uncertain situation?
That's an extremely interesting question, and it's one that's taxing the whole field at the moment. There's so many unknowns about putting stem cells and stem cell based products into human bodies, and so many unknowns about biomaterials, let alone the combinations that we're using. And one approach would be to not really go anywhere near a human being until you knew everything there was to know about the way the cells interact and the cellular mechanisms and the genetics and the immune responses and everything you possibly could in small animals and in the laboratory before you progressed to human trials. But that would take many, many decades. So what do we need to know in order to go into clinical trials? Well, first of all, we need to be aware of the clinical need. And to start with, most of these highly innovative products are going into patient groups where there is no good treatment at the moment. So really you're trying to produce something where there is no other alternative. You also need to know that there's some evidence of potential efficacy, that there's a good rationale for it working scientifically, you can hypothesize how it might work, and that in animals you've shown that it does work, even if you don't know exactly why and what all the mechanisms are. And also in those animals you've found that it's safe, that it doesn't seem to cause toxicity, it doesn't cause cancer, it doesn't break down, it doesn't go to the liver and cause problems there. All of these things. So you then discuss with the regulatory authorities whether they feel you have sufficient evidence, and if not, go back and do further experiments. But there comes a point where patients are needy, you have evidence in animals that it might work and that it's safe, and at that point you can then go back to regulators with a designed clinical trial, usually of only a few patients, a so-called phase 1-2 clinical trial, to test it in those people. There is an intermediate step which we have engaged with a couple of times, which is where you can use one of these treatments on a compassionate basis. So for somebody who is dying and for whom there is no conventional treatment, and on several occasions we have done that. We've used tissue engineered windpipes to save the lives of people. And on 2 occasions it worked very well, on the 3rd occasion it didn't work quite so well. But although these were very one-off in very sick patients, they have given as much evidence, we feel, as some of the animal experiments, these one-off compassionate experiments. And if you put the two together, it's actually good evidence that you could actually see some efficacy in human trials. And if those phase 1-2 clinical trials in a small number of patients support the idea of safety and efficacy in humans instead of animals or one-off ill patients, you can then start to move towards large-scale clinical trials, which really give you the powerful evidence that this is going to be a treatment that's worthwhile. So it is a very fine balance, and some countries are more on the side of accumulating as much science as possible, and some of them are on going into patients quickly. I feel in Britain we probably have the right balance.
Would you say that using induced pluripotent stem cells to develop disease models is a good step to reduce the time it takes in gathering evidence whether it would be efficacious in humans or not?
So induced pluripotent stem cells are a new technology, relatively new, pioneered here and Japan of course, and they have very considerable advantages over a lot of the cells that we're using at the moment. They are already in clinical trials, of course, in Japan, where they're being used for patients with retinal degeneration. And so far, my understanding is that they haven't seen a lot of toxicity. There are concerns about iPS cells, though, about to what extent they might retain some of the memory, as it were, of the original cells from which they came, and also about how uncontrolled they might be. There are some similarities in the generation of iPS cells to the processes whereby normal cells become cancerous.
Do you think using induced pluripotent stem cells to develop disease models could reduce the disparity between animal findings in preclinical research and clinical trials?
So there's been a lot of interest recently in developing much better in vitro models of organs and diseases. And an example would be using scaffolds and cells in bioreactors to mimic liver tissue, for example. And this provides a wonderful opportunity for screening, rapid throughput screening of lots of different molecules or potential drugs or gene therapy or any of the potential therapies that one might envisage. Having got through that rapid screening phase, one would still have to do the animal studies though. They won't be a complete replacement for animal studies, but they should reduce the need. It also means you can use human cells in a human, what we call organotypic model, rather than relying on animal cells in animals. In fact, we have often used human cells in immunosuppressed animals, and one could argue about the relative merits of those various models. It doesn't really matter too much. So what we need to know is just how close to real human tissue we can get in the laboratory in one of these bioreactor models. And one way of doing that would be to use stem cells or induced pluripotent stem cells to generate these tissues. We do need to understand a lot more about the way iPS cells behave in vitro as well as in vivo, and in particular their potential carcinogenic potential and the way that they remember the parent cells from which they came. But they do offer promise, and I think that it will be a very neat bridge. In fact, it may even become a required bridge for regulators going forward before one actually contemplates clinical trials of new therapies.
What is a day in the life of a clinician scientist like?
So it is very varied, and you're constantly juggling because, I mean, certainly if you're a clinician, a surgeon scientist, and some people think that they don't really exist because you can't be a surgeon and a scientist, and the surgeons don't think you can be a proper surgeon if you're doing science, and the scientists don't think you can be a proper scientist. So anyway, that's beside the point. If you're trying to be a surgeon scientist, you have all of the stuff about operating, but the pre-op care and the post-op care and the clinics that go with it and the administration that goes with that and the teaching. But on top of all that, you've still got to be writing the grants and running a large research group. I've got about 10 PhD students or something at the moment. So it really is juggling and prioritising. You can't get too involved in detail. Obviously, when I'm operating, I'm involved in detail. But I mean, otherwise, you have to delegate appropriately and really target your support. And yeah, it's immense fun when you reach this stage, but still very challenging. Getting here, the career pathway to get to this point is very convoluted and extremely difficult, and so one of the things I'm trying to do is to make that easier for future young surgeons wanting to pursue an academic career, to actually make that a lot easier for them, because there's so many roadblocks for them on the way.
So a typical day would start, would you have ward rounds as a surgeon?
So it's hard to say there is a typical day. So Mondays, for example, I do a research clinic, so I just see all the students one after another, like a clinic only with students, and then we have some teaching. And then Tuesdays I would write a lot and have meetings and see a few patients. Wednesdays it's usually clinic all day, generally, with some teaching. Thursdays in theatre all day. And then Fridays again is a mixture of things, often giving lectures and teaching and travelling. So really quite mixed.
That does indeed sound like a full but very meaningful schedule that you have for the week. Makes me feel like wanting to follow you around to see what it's like. But I think that's probably all the time that we have for today.
Thank you very much, Dr. Birchall, for taking time out from your busy schedule to talk to us.
Not at all. Thank you.
So that was Professor Martin Birchall for you all. I'm sure this interview was very insightful for all the aspiring clinician scientists.
If you'd like the transcript for our blog, you can click on this link, and we'll see you next time during our next interview. Bye!
Bye!